Hello, this is the second lecture in molecular genetics. It is on terminology. You probably have some of these terms in your session on medical terminology. However, no harm. Let us refresh your memory. Sorry. Heredity or hereditary. Heredity is the noun, hereditary adverb. Genetics, variation. What we mean by this? So heredity is the transmission of traits from one generation to another. The traits, the physical traits, diseases, anything transmitted from your father, your grandfather, your previous generation, very old. This transmission is heredity, and the disease called hereditary disease, or the trait called hereditary. Okay, variation, it is the difference between you and your brother, and your father, and your son or daughter. So these differences, we call it variation. Genetics is a study, it's science of heredity as a science, and hereditary variation, it's adverb. Okay, this is the genetics, and of course you can study cells, individuals, their offspring, and population, the community. So this is the genetics. Let us have some idea about inheriting biological traits. So the parent, of course, it's coming from a gene, and that genotype expressed in phenotypic traits. So all character, natural talents, even genetic disorder. Of course, we are concentrating on the disorders, the diseases. You will be a doctor. You have, as we said in the first lecture, you will face some of your patient having diseases, and these diseases are related to genetic factor. Okay, let us have a simple question. Let us be aware now from the diseases. So this dimple, you see this dimple? It's beautiful, yeah. That's what you have it from your parents or grandfathers, grandma. Thumb, regular thumb, or irregular tongue. If you can roll it up, this is simple. This is not diseases, but we just give it as an example. So you will inherit these from your previous generation. Okay, genetic engineering. Genetic engineering, it is alteration of the DNA, and use it in medicine, industry, or many other purposes, even in agriculture and forensic medicine. Genetic engineering technique, as we said, we use it in research, in agriculture, industry, biotechnology, medicine, forensic medicine, and so on. For example, or just an example, insulin. People use it to treat the diabetes. You will have it, you will study it more and more in your next academic years. You have the growth hormones. All these could be produced in the laboratory, okay, by genetic engineer techniques. Even groups. Yes, we are concentrating on man, on a human being, but animals, plants, you can use the genetic engineering. This is one we call talking about groups. You won't have a group produced more than usual, more than the normal one. You increase the product. You will have a plant resistant to some environmental factor like heat, like salinity, for example, or diseases, plant diseases. So this particular, by genetic engineering, you will get group resistant to the disease than ordinary one. Okay. As I said, we are just talking about, now just mentioning terminology. We are studying is very much, it's just definition, description, just to understand it. Chromosome. Chromosome, it is a chromatin condensed into compact structure. You know chromosomes. It's like threads. And these are, these threads are composed of chromatin, and it is very condensed. Chromosome, your chromosome in your body, of course you have 46 chromosomes. Some of them sex chromosome and the other are somatic. The sex chromosome is the XY, X and Y, XY in male, XX in female. This is a real one. These chromosomes, this type of picture, they call it karyotyping. So you will take, well, we will study it more, I hope, in your third year in pathology. You will have cell, you will culture the cell, for example, peripheral blood lymphocyte. You culture it, you stimulate it to be cultured, then you harvested the cell and you have prepared the chromosome, then you photograph it and cut it into pieces by scissor, then arrange it from the longest one to the shortest one. Longest one is number 1, shortest in number 22. So why these are numbers? Number according to their length and the position of the centromere. Is this male or female? Of course, this is male. X, Y. And this type, we call it karyotyping, and it is G band. These bands, you treat it with trypsin, certain chemicals, then you stain it, and you have pale and dark bands. Of course, you can, recognize this or prepare it. You need to be an expert to produce this, and it is not your job to prepare. This is somebody else will do it for you, somebody working in the laboratory, so don't worry. We have to divide the labor between us. Some of us treat, diagnose, some prepare the biological material, and everybody is important. Everybody important in your hospital. The cleaner, the staff, the junior staff, the senior, the management, the head of the hospital, all of them important to give a good high standard health care to your patient. So this will be prepared for you and will be diagnosed, and you will get the report. So don't worry, unless you are going to be a cytogenetist, you will decide probably to go on your higher education to study this type of science. If you will be GP or specialized in pediatrics, gynecology, you're not supposed to prepare this yourself. Somebody else will prepare it for you. So don't worry about the difficulty of this. This is karyotyping. What is the difference from the previous slide? This one is just cartoon, just drawing. We draw it as for demonstration. And these are all autosomes, and these are sex chromosomes. And of course, you can't find it in your real life. You can't find it both. It is either XX or XY, but it is, they are put it here just for demonstration. Okay, and you know the chromosome, this is short arm, this is long arm, this is the centromere, and the centromere where the chromosome attached to the spindle. You remember this in your cytology, in your biology. Okay, this will be divided into 2, and each of them will be will duplicate, and we'll have another chromosome. So human being got 46, so your daughter or son will have 46 as well. If they have more or less, they are abnormal. Okay, this is the abnormality. Abnormality of the chromosome could be structural one or could be numerical one. and don't worry about it, we will study it further. Now it is just a term. Deletion, break, this means break, chromosome break, and a piece is lost. Inversion, you have two breaks in one chromosome, and this will be inverted and reunited. Translocation, two chromosomes have a break in these two chromosomes, and piece from A going to B, and from B to A, so they are translocated, crossing over. And these are just demonstration. This is the translocation, and this is the color. It's not real, of course. They are painted in different colors just to show you, to explain it to you. So you have piece from the gray and piece from the pink. The pink one go to the gray, and the gray one comes to the pink, and here you have it. So they are translocated. And why balanced? Because the exact piece coming from this one to the other one. Okay. There are different. Here is the ring, for example. Just an example. Here a fragment. This is wrong. It's called fragments. Must be a fragment, just one. Here is fragments, two fragments. If this chromosome cut into two areas, so we'll have this fragment. And if this unite, it become like a ring. So this is ring chromosome. Of course, ring chromosome. If there are many of them, they call ring chromosomes. Okay, inversion. This piece cut in two. This long arm cut it in two areas, and this piece turn over an opposite side and reunite, so it is inverted, and they call it inversion. Okay, these all are called structural abnormalities. Structural, it is in the structure. You got the same number from, if you have 46, yes, they will be 46. However, they are structurally abnormal. It's not like the original one. Now I have 12 minutes. I have to stop after 1-2 minutes for easy uploading of this lecture for you. And here is the abnormality in the number of the chromosomes. So you remember we said in the first lecture, lecture 1, and we gave example for Down syndrome. You and I supposed to have 46 chromosomes. The poor patient of Down syndrome got 47, and the extra chromosomes 95%. More majority of your patient in with Down syndrome having 47 chromosomes. 5% they have some other abnormality. We'll discuss this in your third year in the pathology, and of course in fifth year in pediatrics. So don't worry, you will have more information on that. However, the chromosomes, the chromosomes called, if it is three set of 3 chromosomes, they call it trisomy, because there are 3 instead of 2. Normal individual have 2 chromosomes, 1 pair from each number. A patient with Down syndrome have 3 chromosomes, and in particular at the number 21. Okay, allele, recessive allele, locus, these terms, what's the meaning? Allele, the different option for a gene. Of course, it's gene, and the gene, if it is different from the same gene, have 2, for example, 2 types, 2 variation. This one called allele, and of course the gene will expressed in a phenotype. And this is example. You see this? This is attached loop of the hair, and hair is separate. It must be there is the chromosomes responsible for this. So gene in a certain chromosome in a certain area. The gene here make it attached, and the gene of the same area in another individual make it separate. Okay, sorry. So it is an alternative form of gene. Allele occur at the same locus on homologous chromosome, and then if you are talking on the plural, you call it loci. Loci. One is locus. Well, this is, I said, this is the loop of the air. Homologous, I think I have to stop in less than 1 minute. Homologous chromosome, homologs, they are member of a pair of chromosomes that are alike. One of them is called homolog. Okay, let me stop here and save it for you. Then we